off we go. So I'm just going to give you an overview of microbial genomics. For those of you studying viruses, I have to apologise because it's going to be primarily bacterial genomics. But many of the same concepts apply, whether you're looking at archaeal genomes, fungal genomes, bacterial genomes. They're all smallish genomes. So here's a few questions that you can sort of think about to prime your mind as to what we're going to talk about in the next few minutes. So how big do you think a bacterial genome is? How do they differ from humans? And what can you learn? You know, if I gave you a bacterial genome sequence, what would that tell you about the bacterium and about its properties? I'm going to draw to some degree on a review I wrote a few years ago of 20 years of bacterial genome sequencing with Nick Lohman, who was a PhD student of mine. So I'm shamelessly going to self-plagiarise some of the images and tables and stuff from there. And you can look it up and read it if you're interested. So the golden era of bacterial genomics, if you like, when we look back and think, oh, that was amazing time to be alive, was when we started getting bacterial genome sequences for the first time. And this began in 1995 with Craig Venter and Ham Smith giving us the Haemophilus influenzae genome and showing us that we could actually get bacterial genome sequences through a process of doing the sequencing, shotgun sequencing, but then assembling those sequences. And I actually was editor of a book that sort of summarised what happened in that period called Bacterial Pathogenomics. And it was particularly a heady and exciting time because we got to see the inner workings of many of humankind's most fearful adversaries for the first time. You know, we could actually see what makes Mycobacterium tuberculosis the thing that it is. What is Yersinia pestis that causes plague? All of this became clear to us. For a few years, every genome was like an Apollo mission or an Artemis mission, as we should say, given the news of today. And they were costing, I think the first ones were getting on for £1 million, they were costing hundreds of thousands of pounds. But that quickly moved on, things moved on, and we started sequencing multiple genomes from each species. You know, we got the second E. coli sequence, the second Staph aureus sequence, and we started to get insights into genomic diversity. And you can see that we were starting to see exponential increase in the number of genome sequences very early on in this particular game. And this is just, as I say, I'm borrowing the figures that we put in that paper. Basically, the revolution came because we could actually do this shotgun sequencing where we fragmented the genome into small fragments, cloned those in plasmids, which we replicated in E. coli, and then sequenced each of those inserts from the plasmids, typically tens of thousands of inserts, and ended up assembling all those to get the genome sequence. And this relied on the method I mentioned yesterday, Sanger shotgun, or Sanger sequencing by synthesis. You make these templates in vivo by growing them in E. coli, which was quite an onerous process. It meant you had to grow those colonies, pick those colonies, prepare plasmids from those colonies. Fortunately, automation did come in, so there were colony picking robots, but I'm sure there were plenty of PhD students who spent many an hour picking colonies in these projects. And that just shows you the timeline of that kind of first era, if you like. There's Fred Sanger down at the bottom, and then anyone recognise this individual? He was the kind of Elon Musk of his day. This is Craig Venter, so he was involved. He was part of the group, headed up the group that sequenced the first bacterial genome, and he also led a human genome sequencing project that was privately funded, and he was seen as the kind of evil man that was threatening the integrity of the publicly funded human genome project. And
was it his genome?
and it was his genome. Yeah. It's a bit like those people that run sperm banks and just fill it up with their own sperm and have hundreds of children. You know, it was his own genome. It was a kind of vanity project in a sense. But yeah, was the irritation in the oyster that gave rise to the pearl? I mean, he did progress the field and probably saved us several years of effort, both in bacterial genomics and in human genomics. Anyway, I'm not going to go through each of these little boxes here. You know, I've mentioned some of these things before. Fred Sanger, the human mitochondrial genome sequence, first genome sequence of free living bacteria. So for those of you who accept the cladistic argument that the human mitochondrial genome is a bacterial genome. And we went on and we sequenced multiple, we showed genome sequences from multiple strains in, for example, E. coli, where we showed that there was extensive horizontal gene transfer. Genome sequencing of multiple strains of Bacillus anthracis. Well, you're going to hear more about that on Friday. I was involved in genome sequencing of the uncultivable Trophorema whippleyi, which led to the design of a growth medium which could support it, so it could be grown afterwards. So that was that first generation, and this is just a box from that paper, which I'm not going to read through it all, but just to prompt me to summarize, you know, there was an interesting sociology. So there were two large centers in the world to begin with. There was the Institute for Genomic Research. It was abbreviated as TIGER, set up by Craig Venter. And there was also the Sanger Centre near Cambridge that was funded by the Wellcome Trust and named after Fred Sanger, as you might expect. But both of them cranked out lots and lots of bacterial genomes, one after another, and there was some friendly rivalry between them. So Tiger beat Sanger to get the first bacterial genome sequence, but Sanger beat Tiger to get the first Mycobacterium tuberculosis genome sequence out. There was a French equivalent called Genoscope, and when we were sequencing Trophorema whippleyi, I was part of the team at the Sanger, working with the Sanger to do that. We beat the French to getting the first genome out. But they then skillfully used the genome sequence, as I say, to design a growth medium for it. And the French team was headed up by a guy called Didier Raoul, who we did approach and say you could join the Sanger team. He said, if I'm not the head of the genome sequencing team, I don't want to be involved. I'll do it in France. And he went off and did it in France. Some of you may recognize his name because during the COVID pandemic, he became one of the bad guys because he was putting down bogus treatments and suggesting that treating yourself with antimalarials would help you overcome COVID. And so again, he's one of those guys who actually has transformed our field, and there'd been multiple moves forward in the field of microbial genomics due to Didier Raoul. But on the other hand, you know, a lot of people think he's a difficult guy. Like, they're both, you know, they're like Elon Musk. You can see there are some advantages, but there's a lot not to like about these individuals as well. Okay, so let's move on. So we got spin-offs from bacterial genomics. So I was involved in the Campylobacter genome sequencing project, and a mate of mine, Brendan Wren, was delighted to find within the bacterial genome something totally unexpected. So what they found was that there were these glycosylation systems within Campylobacter genome, encoded within the Campylobacter genome, that were very similar to the systems used in eukaryotes to glycosylate proteins. And this led to a vibrant program of glycoengineering, which is going on to this day. And in fact, there is a company that Brendan set up called ArcVax, which is delivering on this glycoconjugate vaccine discovery program. Another interesting spin-off was when people started looking at different genomes, they saw that there were these funny things called CRISPRs in there, which stands for clustered regularly interspaced short palindromic repeats. And there were variable arrays of these, like long repeats of these things. And Eugene Koonin, who is another one of he's a giant in the field, and no, there's nothing offensive about him. He's almost like a human computer. His output is just so tremendous. There is an issue that if you go and look him up on PubMed and look at how many papers his name is on per year, you kind of think, have you really read all those papers that your name's been added to? But he is a giant in the field. And he said, hang on, maybe this is some kind of immune system that the bacteria are using to protect themselves against bacteriophages. And those insights primed the exploitation of those systems by Jennifer Doudna and others to use CRISPR-Cas as a genome engineering technology. And so, there were things that we expected to discover from bacterial genomes when we sequenced them. We wanted, where are the toxins and the adhesins and so forth. But there were other spin-offs as well. And it did prime these advances. This is a slide from a review by David Relman, who's another one of the leaders in the field that I collaborated with on the Trophonium Whipple project, just showing you the kind of uses that you could make of a genome sequence. So you could look at the population structure and evolutionary history of the pathogen you're interested in. You could investigate outbreaks and strains and so forth. We'll say more about that. We already mentioned you can look at growth requirements, you can devise new diagnostics. So you've got the whole genome or you've got multiple genomes, you can then devise a simple PCR test that would allow you to detect that thing or to type that particular organism. You can raid the genome for protein coding genes and then get out the proteins and express them in expression systems to allow you to make new diagnostic targets or new vaccine targets. And you can also, by getting insights into those protein targets within the cell, you can design new drugs that might target vulnerable parts of the bacterial system. And also, we get insights into how bacteria cause disease. And we're speaking later in the week about pathogen biology. What does it take to be a pathogen? Our understanding was greatly informed, has been greatly informed by having genome sequences. And here's just a few of the early ones that I look back on with a sense of nostalgia and pride. The first Campylobacter jejuni genome sequence, the first sequence of Corynebacterium diphtheriae. That was an interesting story there because at the time that we wanted to start that project, there was an outbreak of diphtheria that was raging across the former Soviet Union, and particularly in Russia. And there were two major research centers in Russia, one in St. Petersburg and one in Moscow, and there was fierce rivalry between them. And it was unclear which should you collaborate with to get a strain that would give you insights into that outbreak. And luckily they managed to come up with a workaround, which was there was an individual, I think, had been on sex tourism to Russia and had come back with diphtheria. I think, if I remember correctly, diagnosed in Newcastle. And so it was a strain that represented the outbreak, but you didn't have to ask for any permission from any Russians, and they sequenced that one. And I mentioned Trophonium whipple, which is a very strange organism. In certain susceptible individuals, it grows in the small intestine silently and takes over all the tissues and actually damages the integrity of the small intestine, so you can no longer absorb nutrients, and people start to waste away. It can also get into the bloodstream and affect the heart valves and cause disease. But the thing was, you couldn't grow it on agar. Nobody could worked out how to grow it. Didier Raoult, as I say, one of these people that did make great steps forward, he managed to grow it in cell culture in association with human cells. But it was only after the genome sequence had come out where he reverse engineered, it needs this amino acid, needs that, and that they actually came up with what we call an axenic medium. It didn't require any human cells in the mix as well. And strangely, during lockdown, one of my primary school friends got in touch with me after 50 years since I'd last seen him. And it turned out he had been infected with Trophonium whipple and had to have a heart valve replacement just a few years ago. And just a few months ago, he had a relapse. So it brought home to me the importance of this particular pathogen. It's a rare organism, but when it happens, it can be devastating. So towards the end of that kind of golden era, I wrote a review for Nature with Brendan Wren, and we basically said that, we've started to get now. We had representatives of every major pathogen of humans, plants, and animals, and we could start to see the forces that were shaping bacterial evolution and genome dynamics. So here are just some of them here. You know, often you see genome duplication, and I mentioned the idea of perology. You see that kind of thing going on. You see horizontal transfer as a big theme. So when we looked at the first, when we had the first E. coli genome, then we got the second one and the third one quite quickly afterwards, and we realised that the actual gene set that they all shared in common was only about 30 or 40% of the genome, and that E. coli engages in rampant horizontal gene transfer, and it's taking in genes from other organisms, very, very commonly. Many of them sit on plasmids or phages for a while, but they get integrated into the chromosome and then become part of that lineage. Other interesting phenomena that we saw that once things move into intracellular niches, so some bacteria like chlamydia or rickettsia move into cells, become intracellular pathogens, they tend to shed a lot of their genome because they don't need it anymore, because they're basically scavenging stuff from the host cell. And the mitochondrion is the ultimate example of that. So the mitochondrion entered a host cell 1 billion or more years ago and threw away almost all the genes that it needed to survive. I think, if I remember correctly, there were 17 protein coding genes in the human mitochondrial genome. So that, we just saw all these things. What we saw also was that when a thing shifts its niche, so if it's been living as a free living organism fighting for existence out there in soil or in the environment, and then it suddenly moves, Microbacterium lepra is an example where it only lives in humans. Well, it only lives in humans and a few animals, it turns out. And it lives inside the cells there. It's actually got lots and lots of what we call pseudogenes, where you can see that there's a sequence there, but the gene has been disrupted. Either it's got frame shifts in it, or it's got SNPs that basically inactivate it, or insertion sequences have jumped into it and disrupted it, or there have been deletions that take out parts of it. And what we found in Microbacterium leprae, for example, and the same also true in Yersinia pestis, is that there was just like this graveyard of dead genes in these organisms. And that was kind of unexpected to see quite that much of it going on there. We saw all these recombination and rearrangement going on. We saw SNPs, single nucleotide polymorphisms, and we also noticed that what we were calling species, the term was a little bit kind of flaky, if you like, because there were some species that looked highly variable and had all sorts of different things going, like E. coli, huge amounts of variability. But Yersinia pestis is one example. Microbacterium tuberculosis is another. These organisms have a very restricted population diversity, and they're effectively a single kind of clonal lineage that has adapted to cause disease and has a particular disease-associated phenotype, and that led us to start to understand the evolution of these things. So here's just a few examples of mobile genetic elements that we know were encoding important virulence factors and are present in human pathogens. So you've probably heard about plasmids and phages. Plasmids, there's not really much more to say about them. They are generally small genetic elements separate from the chromosome that replicate independently of the chromosome. Generally they're circular, although you can get linear plasmids. Phages are interesting because phages are like bacterial viruses that can come in and infect the cell and cause havoc, take over the cell, make many copies of themselves. But they can also become what we call prophages, where they can integrate their genome into the genome, the chromosome of the pathogen. And in that context, they often bring in useful genes for the host. So it's kind of a bit paradoxical, like, who's benefiting from this here? Is it the phage is looking after itself, or is it looking after its host? And many of these Prophages are carrying important virulence genes. And the other key idea that came along was this idea of pathogenicity islands, so that when you looked at genomes, you could often see there's a whole chunk of genes, dozens of them in a row, in taking the organism up to a new phenotype, like a quantum leap towards a new phenotype. And these genes appear to have been acquired by horizontal gene transfer. Unlike with plasmid-coded genes and prophage genes, there's no smoking gun usually as to say, well, how did they actually get into the genome? But they're generally, if you look at genome comparisons between multiple strains within the same genome, or you look between genomes from closely related species, you can see these things have kind of come in, wedged themselves into the genome. And one of the things that we started to sort of have to think about, and again, this is, I say this every now and then when we look at conceptual background stuff. You're doing a PhD, a doctorate of philosophy, and sometimes thinking in the round about the concepts that you're dealing with is important. And it is the sort of thing that an examiner can pick you up on. If they could say, well, tell me, what do you think a virulence factor is? And they can ask you that, as the opening gambit in your PhD viva. And if you've never thought about it and you don't care, you can go into a terminal tailspin straight away, and then the rest of your viva doesn't go so well.
I don't know the answer.
There is no answer. It all depends on what direction you're looking at the thing from. And there's that old, I think in England we say it comes from Sri Lanka, this idea that you've got the blind men looking at the elephant, and one of them feels the trunk and says, oh, it's like a snake, and the other one feels the tail and says it's like this, and it's a bit like that with defining a virulence factor. It all depends on what direction you're coming from and how you're looking at it. So what we started with in bacteriology is Koch's postulate. So Robert Koch said, look, okay, if we want to show this thing's a pathogen, we stick it in an animal model. We see that it causes disease in that animal model. It's present only in people who've got disease, not in healthy individuals. And we can grow it from those diseases. Then we realised that wasn't quite so simple. So that, if you've got a salmonella outbreak, 100 people have eaten food infested with salmonella, 50% of them or 20% of them might get obvious clinical disease. But if you went and cultured the stool samples from the rest of them, there may be a considerable number of them that don't have any clinical symptoms, but the salmonella has passed through their system, it's colonised their gut for a short while. And that's the case across the whole spectrum of disease pretty much. That subclinical infection, which can be detected either by looking and seeing the thing there, or often you make an immune response even though you're not even aware you've been ill. These are common things, so that undermines it. There's a guy, Stanley Falco, another one of the greats in the field. He said, well, what we can do is we can adapt Koch's postulates. If we want to find out what a virulence factor is, what we do is we see that virulence factor is present in all the pathogenic varieties of a strain and not in the non-pathogenic varieties. We knock that gene out, and this was building on the new tools of genetic engineering to knock a gene out, and we can see in the lab, that particular virulence factor disappears, or we can see in an animal model, it's no longer capable of causing disease. And the interesting point that Stan included in that definition was he said, and then you have to put it back in again and show that you get back to virulence. And what he's highlighting there is that if you take a bacterial strain and grow it in the lab repeatedly, its characteristics will change. It will acquire mutations as it adapts to living in the lab. And so if you see a change in phenotype, you can't be absolutely certain it's because you've knocked out a gene. It could be there's another mutation somewhere else in the genome that's led to the loss of. So what we call complementation, you put that gene back in again and you show, we've got that back again. But again, there are difficulties there because if we're looking at, say, E. coli causing disease, first of all, it has to survive in the gut in the first place before it can actually start disrupting the gut and causing disease. And all the things it's using to survive in the gut, or most of them at least, will be shared by the commensal E. coli that we're not saying are pathogens. So are we saying that the things it requires to adhere to the cell, to the lining of the gut, those are all virulence factors? Under the molecular Koch postulates, you'd say they are. If you knock them out, then it can't cause disease anymore. But they're shared with the commensals. So it's all starts to become a bit blurred, actually. And so it depends on what you're looking at. You can use comparative genomics, you can use mutagenesis, many different ways of defining a pathogen. And in this paper, we said actually you've got to come up with an eco-evo view, an ecological and evolutionary view of virulence. So another key insight is that things that look like they are pathogenicity factors, like they're a toxin or something like that, why are they there in some commensals or in environmental organisms, as well as in things that we say, oh, that's definitely a pathogen? And one insight that became clear is that bacteria are involved in the struggle for life, to use Darwin's term, and they're struggling not just with humans in causing disease in us, but they are struggling in their interactions with phage, with other bacteria, with microscopic eukaryotes, with invertebrates, with plants and animals, fungi, and so forth. And so those virulence factors may be sitting there because they're useful in a particular ecological context. So one key example is that Legionnaire's disease, you catch that typically from breathing in water droplets. It lives in air conditioning systems. It also lives in natural water systems as well, waterfalls, anywhere where you get an aerosolization. Gets into your lung and starts to cause disease. But it's tooled up with all these virulence factors because it interacts with amoebae in its natural existence. And once it meets a macrophage, it just sort of says, oh, you look like an amoeba to me, and it deploys all the same things it would deploy with amoebae. And in fact, Brendan Wren, who I mentioned earlier, they're just doing some interesting work at the moment showing that Campylobacter jejuni can live within amoebae as well. And so this kind of ecological theme of looking across and seeing what's going on in the ecological context. These are interactions between bacteria and eukaryotes, but then maybe not maybe not multicellular eukaryotes, or maybe unicellular ones. Another thing we noticed was this idea that basically there are switches in lifestyle, that things can be free living, they can be commensals, they can become pathogenic, but then they may lose a pathogenicity island or whatever and go back to being commensals again. One of the things that we discovered when we looked at even at E. coli, so people had this idea that E. coli is like the archetypal organism and it's all perfectly designed to be E. coli. And then you look in it and you find there are all sorts of broken systems with bits of rubbish, genetic rubbish lying around. And this is clear that E. coli has in the past deployed, say, secretion systems or motility systems that it's no longer using, but you can see the evidence is present in their genomes. And so this is just a slide that just shows you some examples of those kinds of virulent systems that are in pathogenic and non-pathogenic bacteria. And we'll say more about virulent systems later. Anyway, let's move swiftly on now to the next era. So that was the golden era where we got lots of insights into pathogen biology. But from 2007, we basically had this miracle of a disruptive technology that basically an utter game changer. So we kind of just got used to the idea, we can sequence genomes. But suddenly we had this technology that took us 1,000 times faster, 1,000 times cheaper. Within a year or two of these new high throughput sequencing approaches arriving, they even became scalable to the average lab. So we got these benchtop sequencing platforms, like the Illumina MySeq, for example. And these really were cheap enough to have and quick enough to have a real impact on real-world clinical problems, but also to become just part and parcel of the average molecular microbiology lab's setup. So I remember talking to one of my colleagues as the business about complementation, or like you've made a knockout in a gene, how do you know it's actually Stan Falco's approach was complementation. But the other thing you can do is, of course, just sequence the genome and compare the genome of your derived knockout with the parent and say there is no difference anywhere in the genome apart from the gene we knocked out. When we first suggested that, we were like laughing. Oh, nobody, it's so expensive and difficult and takes so much sophistication to sequence the genome. Nobody would be doing that, but it is now routine practice and everyone does it. And if you didn't do it and you put a paper in, someone would say, why haven't you sequenced the genome to show it is what you think it is? So it's amazing that the way things have moved on. And this is another part of the figure there. I'd say you've come across the Illumina sequencing and this wonderful idea where you have this bridge amplification going on. And this, during that period, we started doing bacterial genomic epidemiology. We started to explore gene function globally using transposon-based approaches. So later, in the last week of this course, you'll come across an approach called TRADIS, where you can basically knock out every gene in the genome and sample what phenotypic effects you get. And not just every gene, but you can sample insertions every few base pairs within the genome. So a tremendous approach. Then we started to see metagenomics coming in, routine genome sequencing in the clinical arena. So now in the UK, all Mycobacterium tuberculosis genomes are routinely sequenced as part of the clinical workflow. So you can track the spread, you can look at the population structure, but you can also detect antibiotic resistance really quickly and easily, much more quickly and easily using genomics than you could using conventional approaches. And of course, there are political things going on. So genome sequencing actually showed that an outbreak of cholera in Haiti was actually brought in by Nepalese UN peacekeepers. So the political undercurrents there, these were people coming in to try and keep the peace. It was an international body sending people in there, but actually caused more, I don't know if they caused more trouble. They probably did cause more harm than good because they brought in cholera, which led to a devastating outbreak. Oh, go away. I don't know why it's doing that to me. So I'm not going to go through this because you did this with Dave last week, but it is basically what we've done here. We swapped out all those plasmids and E. coli picking and stuff, and we instead have molecular colonies that we grow in vitro. One of the things that I got involved in during that period was we did a performance comparison of benchtop sequencing platforms, and we showed that the Illumina MiSeq gave the best accuracy. The other two platforms had a problem where they had a particular problem where there's what we call homopolymers. So if you've got 8 A's in the genome, the systems can't tell whether you've got 8 A's, 9 A's, 10 A's, and you get errors associated with those homopolymers. Whereas Illumina was much more accurate for those homopolymers. So in fact, the Illumina reps used to give out our paper when they were trying to get people to sell, and should have bought shares in the company, I suppose. The third revolution was the advent of single molecule sequencing. PacBio with this so-called smart sequencing by synthesis. We don't have that here. They have it at the Earlham. It's one of those technologies. There's a strange dynamic, a competition between Oxford Nanopore and PacBio for long-read sequencing. The form factor of the PacBio machines, they're large instruments, they're quite sophisticated, a lot of maintenance required. But they probably are they are more versatile than Nanopore. They're slightly more advanced in some ways. But obviously with Nanopore, the form factor is a little handheld device there, palm top sequencer that can be deployed out in the field. Next year I've arranged for Josh Quick to come and give you a talk about his experiences going to West Africa during the Ebola outbreak and taking the lab that he used for sequencing out in his suitcase because he took out the MinIRON with him. I'm anticipating what I was going to tell you. There's actually a YouTube talk. We don't look at the YouTube talk because he's coming. I think we've got him lined up for a February masterclass with you. And of course, all of this, we have been at the forefront here in Dave, Andrew, Justin O'Grady, who used to work here. He still works in this building, but he flipped over and he now works for Oxford Nanopore. So, and the Bill held the weight of the world on his shoulders during the COVID pandemic by analysing all these sequences that came out of these efforts. So this is a thing.
Sequenced 80,000 COVID samples.
Yeah, 80,000, which is.
And we were sending reports to government and doing all sorts of things and watching all the politicians ignore it.
Yeah. It was exciting. It was one of those times where all this groundwork of trying to understand how to do these things paid off, both in terms of the lab side, but also the bioinformatics. Right, I'm just going to say a few. Well, let me just pause for a second there. Has anyone got any comments or questions about the stuff we've just spoken about? You can all moan and say, what are you telling us about history for? You know, why don't you just tell us how it is right now? And there's some truth in that, but there are also some lessons from history as well about how things are played out. Okay, I'm just going to take you through a few quick slides now just on general features of genomes to lay the groundwork for the work you're going to do this week and in subsequent weeks. So you probably don't need me to tell you this, but microbial genomes are quite different from the human genome. And there's quite a gear change needed if you move from one to the other. We've got small, what you see is what you get kind of genome. So basically bacterial genomes, most of what's in there is coding for proteins. That's quite different from the human genome, where there are huge stretches of the genome that don't code for anything. There are all these huge great introns and all sorts of stuff going on. You do get introns in bacteria, but they're very rare. We did actually sequence one of the first introns back in about 1990, I think, but they're extremely uncommon. The protein coding genes, because it's what you see is what you get, they're typically very short and compact. The average protein coding gene is about a kilobase, about 300, comes out about 300 amino acids is the kind of average. And we have these operons where we've got promoters just upstream. So the genes are clustered together in operons where they tend to have related functions. They may be enzymes all in the same pathway, or there might be a transporter for a substrate and then an enzyme that uses it or whatever. I have to qualify that slightly to say that all this concentration on operons is because we've used E. coli as the model organism and we have an E. coli-centric view of the world. When we first sequenced the Campylobacter jejuni, we discovered that it didn't really have operons in the same way that E. coli does, and it wasn't quite so easy to read the sense as to what's going on there. And there are fewer non-coding RNAs. There are non-coding RNAs. We have the tRNAs and ribosomal RNAs. There are, though, now it's clear there is all this cross-chatter with non-coding RNAs in bacteria, but there's a lot more that goes on in the human stuff. And we mentioned chromosomes and plasmids. Most of the time these are circular, but you can even have linear chromosomes in some organisms. And a few organisms, Vibrio cholera is one example, you can have 2 chromosomes. Plasmids, you can have variable numbers, you can have 1, low copy numbers, you can have 10s or more. Often their copy number is tightly regulated so that they're in a particular proportion to the host chromosome. And they often carry non-essential genes that confer an adaptive advantage in certain conditions. How do we do the sequencing? I've just mashed together various talks here, so some of this we've covered. But basically the idea of shotgun is you just rip the thing into pieces. and then you do the jigsaw puzzle of putting it all back together again. This requires sequence assembly, so you can look at bits of sequence that overlap. Where it's clear that they overlap and there's no ambiguities, you just say, okay, we can build a larger sequence, which we call a contig from that. The big snag is where there are repetitive regions in the genome where it's not clear. So a bit like when you're doing the jigsaw and you've got two identical pieces of jigsaw that just have blue sky in them. And so it is these repeats which are one of the confounding issues when you're trying to do assembly. And you do this assembly, we often do paired end sequencing, so we sequencing both ends of a fragment, and we can put all these together into contigs. And it may be that we don't actually have a sequence for a particular, we just don't have that good enough sequence to straddle a gap. Sometimes the sequence might not actually be present even in maybe just, you have to get to a certain depth of coverage to be certain that you've covered everything just by chance. So if you go in with low depth of coverage, you might just get a dropout because it's not there. But another problem you get is a physical gap. And this used to be a big problem in the old days when we cloned in E. coli, because there were certain pieces of DNA that when you cloned them in a plasmid E. coli, they were toxic. So they could not be cloned. Luckily, with the methods we're using that are now using in vitro approaches, that's not so much of an issue. Okay, once you've assembled a genome, you do annotation. You basically go through and look at all the predicted sequence features. So this idea, if you look at the GenBank format, the difference between GenBank format and, say, FASTA format is it doesn't just say here's a sequence and here's a header. It annotates various features and says, oh, there's a feature starts at position 2 and ends at position 25 or whatever. And the way GenBank format works, all those features have a standard kind of nomenclature and a way in which they're encoded in the file format. And basically we want to add all those features. And one of the things that's most important is identifying the coding sequences. Coding sequences means the protein coding genes. You often see the abbreviation CDS for those. And a little tip, again, because you may get a grumpy old bastard professor in your viva. Some people use the term ORF as a synonym of coding sequence, but in fact they're not synonyms. And we're going to see this in Artemis soon, that you can have, all an ORF is an open reading frame. So it's just a stretch of sequence without a stop codon in. And in bacteria, you can often have multiple overlapping ORFs in different reading frames that expand for a certain amount of time, a certain amount of sequence. And only one of those may actually code for a protein coding gene. And the others are just spurious. They have no biological meaning. And they will not be translated. And the other thing is, of course, that the translation only starts at that start methionine. The open reading frame that corresponds to a coding sequence may have an upstream region that is never translated, but there's no stop codons in it either. So yeah, we use homology searches, we look at all these other things. One interesting thing that we discovered when we started sequencing genomes is you can actually locate the origin of replication by looking at a thing called GC skew. You can calculate percentages of the numbers of bases, the genes, and all that. We'll be seeing some of that later.
Does anyone know what it would look like? How you find it? So Mark's talking about the bias of the GC content, or the coverage as well will change.
Yeah.
Sequence coverage will change. What would you expect it to be?
Yeah, this is a weirdness actually when you look at how much of what's the gene dosage of a particular gene in a bacterial population. The ones that are closest to the origin of replication tend to be overrepresented.
You should know this coming in Gemma's group. That's all they talk about.
Well, this is, yeah, exactly. Gemma makes a whole living out of this issue, but yeah, all right. I'm cognizant of the fact that I keep.
Back to it later.
Yeah. It's fun. I mean, I'm just trying. I'm thinking that I'm reminding you of stuff that you kind of knew, and not give it. So I'm going to try and go fast through this stuff. Am I going at the right pace, or you want me to go a bit faster?
I think you can go a bit faster.
Okay. So mapping is this idea of assembling the jigsaw. It looks at alignments. You got this kind of thing here. SNP calling is where we look for SNPs. Some people call them single nucleotide variants, and again, it's one of those things where people get very pedantic, and I never quite understood what the pedant. But these can be used to identify biologically significant changes through the results of natural selection. But as we pointed out yesterday, you can actually get neutral changes. Much of bacterial evolution is neutral. You just get one turns into another, but you can use that information. And then just these are just a few screen dumps showing you the kinds of things that we could start to do once we got lots and lots of genomes. So this is a genomic encyclopedia of bacteria and archaea. So you can actually start to look at the great tree of life and put everything in its place. And this is continuing now. There's a thing called the Genome Taxonomy Database, which takes all genomes of bacteria, classifies them into species, but doesn't just do that. It takes them all the way up to phylum level, the whole hierarchy, everything in its place. Huge efforts gone into coverage of the human microbiome and getting a reference set for that. We've even gone into the past and sequenced bacteria from hundreds of years ago. So a draft genome from Yersinia pestis from the victims of the Black Death. This was a jaw-dropping moment. Wow. We actually have shown not just that Yersinia pestis was the thing that caused the Black Death, because there were some questions about that, but we actually got the genome of the organism that caused it. These are slides from a talk I gave a few years ago. The world has moved on tremendously since then, and I could give you a whole hour's talk about the evolution of Yersinia pestis since the time of the Neolithic to the modern day. I need to charge my battery up soon. I mean, this is just one weird thing that you can do with DNA. You can actually, this guy Ewan Burney, who's a very bright chap who works at EBI, said, why don't we actually use DNA? We can make DNA now. Why don't we just make DNA and encode stuff in it? So, you can stick the works of Shakespeare, or you can take a photograph and encode it in DNA. And he made the point, actually, this is quite a robust storage system. I've put that on a solid surface there as DNA. That's going to last for thousands of years, far longer than the vellum that they used to write laws in the House of Commons or whatever. And it's one of those bizarre ideas. Whether it will actually grow legs and go anywhere, I don't know. But it just shows you once genome sequencing becomes too cheap to meter, which is more or less where we're at now. Sequencing DNA is so cheap that this is where you end up. I'm going to try and plug in, I think, because I've got 5 more minutes.
Someone tried this out by encoding the password for Bitcoin in DNA.
Really?
Yeah, and then you had to get the money, you had to and then encode it.
Yeah. So this is the mad world we're in, where we've gone from a genome sequence being the equivalent of Apollo mission to the equivalent of stop it! I didn't want to go on eduroam. Of buying a cup of coffee in the canteen. Here's some of the birth of genomic epidemiology, where we could sequence loads of things. Oh, stop it! Just cancel. Go away. I don't want to go on eduroam. And I mentioned Haiti. We're going to talk about anthrax, but one of the weirdest things was they studied leprosy in America. So Mycobacterium leprae, as I mentioned, generally was thought to be an exclusively human pathogen that lived intracellularly, spread from human to human. Does anyone know what the other common, long recognized host was? It turned up in the 9-banded armadillo. And the 9-banded armadillo turned out to be a useful laboratory host. And I have this vision when they sequence the Mycobacterium leprae genome. They effectively put the organs of an armadillo into a blender to get enough biomass to be able to sequence it. But in America, Mycobacterium leprae not only existed in humans, but in the southern United States, it escaped into the wild armadillo population and became a problem in armadillos. And what they showed by genome sequencing was that the leprosy in the armadillos was a subpopulation of leprosy in humans. So that basically this is what we call an anthropognosis. Somehow or other, those armadillos caught leprosy from humans, in contact with humans. But it gets even weirder, because then they discovered there were a few cases in the southern United States of people who caught leprosy who'd been eating armadillos. And there is this thing that, people eat roadkill in the US, and they were able to show an epidemiological link between leprosy in armadillos and then coming back into humans. So it's just kind of does your head in. I know we need to move on, but I will share one. When you get to my age, you don't get surprised by much. You've seen it all. But a few years ago, I was knocked off my chair. Does anyone know where else leprosy lives in the living world? This was something that I just I could not believe it. It lives in squirrels. It lives in red squirrels and grey squirrels. And there are two kinds of leprosy that have now been described that have very similar presentations, but the organisms that cause them are actually a few million years apart. One called Microbacterium lepromatosis. Both of them live in squirrels here in England and in Ireland. So leprosy's been out there in the wild, living under our noses, and we didn't even know it was there. It's just amazing. Oh, go away. This is just basically to explain it's perhaps a bit condescending. I borrowed these from Jennifer Gardy, but it's the idea that you basically, as time goes on, you acquire these SNPs. You can then look at these kind of patterns and look at interactions between populations. You can use this to confer. Oh, all right, show me the certificate. Okay, continue. Oh, crying out. Sometimes computers just don't know who's in charge, do they? Trying to help me, but it's not really helping me. These are sort of the ways in which you can use whole genome sequencing outbreaks. I'm going to pass through all these quickly. And I'm just going to spend 5 minutes, if you forgive me, with one example from my own research, just to give you a tangible example of how we've used genomic epidemiology. So Acinetobacter baumannii is a gram-negative bacterium. It's multi-drug resistant. It's moving towards a situation where we have almost no agents left to treat it. And it's associated with wound infections, ventilator-associated pneumonia. It's common in hospital infection. It's a hospital organism. You don't tend to see it out in the community. It causes bloodstream infections. But one strange feature is that it's very commonly seen, well, not commonly seen, it's recognisably seen in military personnel returning from combat zones. And when UK was involved in the wars in Iraq and Afghanistan, we saw soldiers coming back from the battlefield with this organism. The thing is, it's quite hard to identify in the clinical laboratory, but we knew at the time that we did this study about 10 years ago that there were outbreaks that were going on in hospitals, but we didn't actually know in detail. We could get a general idea, they're all the same strain, so this is an outbreak. But we couldn't say, that patient gave it to that patient, or this ward got it from that ward, because we weren't getting enough fine-grained resolution. And so we turned to using genome sequencing. So there was this outbreak in the hospital I used to work with in Birmingham. The first case was in a military patient in July, and that was typed by what we now call UKHSA. And they gave it what was called a pulsar-type, using a very old-fashioned technology to sequence type it, and we had an outbreak that lasted for 80 weeks, involved 60 patients, involved military and civilians. And what we did was we gathered up 91 isolates of the bacterium from patients and from the environment, and we genome sequenced them on the Illumina MiSeq. And what we did was we often sampled the same patient multiple times as well from multiple sites. And what we found was the first thing we discovered when we got the first genomes out, we said, well, this strain looks quite distinct from any other genome sequence strain out there. It is a distinct outbreak going on here. And we wrote this up if you're interested to look at the paper. It came out in 2014. And one of the things that was most striking was it did make sense, that we could actually segregate out by looking at the whole pattern of SNPs within the genome, the isolates into a set of different genotypes. And that the genotypes actually, as time went on, they acquired additional SNPs. But they had, if we took the first isolate, we compared it with later, not much later, they had a few SNPs. You go a bit later, those SNPs are still there, but a load more SNPs are coming. And so it did actually make sense. We didn't know that it was going to make sense when we started the project, but it all came together and actually made some sense there. So the numbers in red there represent the SNPs, or the SNVs as we call them. P indicates loss of a plasmid. And what we did, when we mapped those genotypes onto the wards, we could see that there was also a pattern here, which we didn't know this was going to be the case, but it was very gratifying to see that we could actually make sense of these genomes. So it started off in Ward 1 primarily. Then it started to spread to other wards, but the odd spread here and there. And this is because the patients themselves move from one ward to another and take the isolates with them, their infection with them. But in some cases, though, it then spreads on in the new context. And the clear signal we saw was that it moved from Ward 1, being predominantly there, to moving into the Burns unit. Now, this was a bit odd because the patients on the Burns unit, they're each in individual rooms with their own ensuite toilet facilities and so forth. They don't go out and chat to each other, share each other's newspapers or anything like that. They are all effectively in isolation. And so it was a very strange finding to see that there was this apparent transmission going on within the Burns unit. But the thing was that because we had the genome sequences, we could say, no, it's not just like random variation of Acinetobacter just popping up from God knows where. This is that very same strain is moving in front of our eyes. What is going on here? We went into the Burns unit and we actually said, what's the common factor? Oh, they all go to a special Burns unit operating theatre to have their wounds debrided. That's the common factor. We went in there, we sampled the environment in that operating theatre, and we found the outbreak strain in the environment there. So it was clear that that was what was going on. And with a deep clean of that unit, we largely, not entirely, but we largely got on control of the outbreak and it disappeared. Oh, this is just focusing on that Burns unit. And you can see those triangles there are the common factor there, that there's a visit to the Burns unit. And that's me finished, and now I pass on. Have anyone got any comments or questions?
Yeah, just really quickly, in the figure you just showed, what were the numbers running down? Is that like patient numbers?
Those are patient numbers, yeah. Yeah, and it's one of those things that the patient numbers were just applied serially to the patients, and it turned out that some of the later patients that had later numbers had actually acquired their infections earlier, or had long-standing infections and so forth. It was a very interesting exercise because you could not interpret those genomes just on their own without any clinical metadata and epidemiological data. But having the genomes certainly gave us a much more insight than we would have got from simply looking at the epidemiology alone. And there were cases that we discovered where there were other Acinetobacter strains in the hospital. So we could say, oh, that patient suddenly got Acinetobacter, they're on a ward where we've never seen it before. Oh, is the outbreak spreading? No, it's a completely different Acinetobacter, and it looks like they brought that in from Luton or Romford or whether other hospital they were in beforehand. So it's interesting. Anyway, that's it. I've had my hour of shedding wisdom.
As a tip for genome epidemiology, generally genomics tends to rule out, which is for the second case Mark was saying, that you see a bunch of cases and you say, well, these are unrelated because they're so distant from the other.
Well, that's the thing, is to prove that things are unrelated, that's a provable thing. To show that they are indistinguishable just means they're indistinguishable, but that doesn't, what does that?
Transmission.
Yeah.
You always need to have transmission. So you might have an identical